And so I'm going to spend most of our time talking about plants because that seems to be a key interest here on the Davis campus. There's a lot of plant biochemistry. But I also need to talk about these purple bacteria and green bacteria from the context that it looks like the systems in plants evolved from these two types of bacteria somewhere in evolution merging together. And so that's the important part there. So here's a cute little picture. Energy from the sun is driving a series of reactions here that historically were called the light reactions, and down here the dark reactions. But the dark term has been replaced by carbon assimilation reactions, acknowledging that these reactions can occur when the sun's out shining also. So in this, we see that when you take a step back, that these two sets of reactions, the light reactions involving photosystems 1 and 2, and these carbon assimilation reactions referred to as the Calvin cycle, named after Professor Calvin at UC Berkeley. He passed away many years ago, but he's credited with doing much of the work here. But there's two sources of energy. ATP, that we're familiar with, is a source of chemical energy that can be used to do things in cells. And NADPH, this is the cousin of NADH. So it has an extra phosphate group on it, but that phosphate group has a -2 charge, and the enzymes that use NADPH are never confused with NADH. But think of NADPH as a source of reducing power. So you've got two sources of energy here, and the pool size in the chloroplast, because that's where all this takes place, the pool size of these things are small. And so if a cell, a plant, or a microbe that does photosynthesis, if it wants to do more carbon assimilation reactions, it's got to break down these two things so that these things can be reduced again via the energy of the sun. So there's this constantly recycling. So another way of saying that is that the light reactions, photosystems 1 and 2, are tightly coupled, very efficient with the Calvin cycle down here, as you might expect. There's no slop, okay? To run this reaction, you need 6 NADPHs for every 9 ATP. So I need you to know that ratio. That's a 3-to-2 ratio. You need 3 ATPs for every 2 NADPHs. So the light reactions produce these things in these ratios, and the carbon assimilation reactions consume them in precisely those ratios. Now, in this process, 3 carbon dioxides from the atmosphere are reduced to the level of a sugar, carbohydrate. So 3 CO2s and a water molecule go to make an old friend of yours, glyceraldehyde 3-phosphate, G3P. Where have you seen that before? In glycolysis. Okay. So G3P is going to be made in the chloroplast, whereas glycolysis is out in the cytosol. And what we'll see is that several enzymes from glycolysis work in the chloroplast, but they're isozymes. They're different forms of the enzyme, presumably because the environment inside a chloroplast is not identical to the environment in the cytosol. pH is different, there's different compounds floating around. Are you with me on that? So life, again, takes something and just tweaks it, apparently, to function better in a certain environment. So this is what's happening here, is there are photosystems, 1 and 2, harvesting photons of light from the sun. Water is being consumed, and in this process, oxygen is given off. And these components are used to drive again the Calvin cycle, which reduces CO2 to a sugar. And then the plant, or whatever organism, can use that glyceraldehyde 3-phosphate. We'll talk more about how it's used in the Calvin cycle, most likely on Friday. Okay. So here is an artist's rendition of a chloroplast. And what they are doing is cutting it in half. And so you can see that there are these thylakoid membranes. These are bilayer membranes. And this is where photosystems 1 and 2 are embedded in these membranes. Thousands of these are embedded. PS1, photosystem 1, PS2, photosystem 2, thousands of copies of each are embedded in those membranes. Out here is the stroma, the liquid that bathes on the outside these membranes, and the stroma is where the Calvin cycle occurs, or what we sometimes call carbon dioxide fixation reactions. CO2 is trying to be fixed, which means covalently attached to something, and then a series of reactions are done that consume ATP and NADPH to make a sugar. So those reactions are occurring out here. And then there's a liquid inside these thylakoid membranes. And I mention that because we really have 3 phases then. We have the liquid inside, which means there needs to be a carrier inside, something that carries things from one position to another. There's a liquid out here, so there has to be a carrier that carries things out here. And lastly, in the membranes themselves, remember the membrane bilayer has a hydrophobic interior. So there has to be a hydrophobic molecule zipping around inside the bilayer, just like coenzyme Q was zipping around, imprisoned inside the inner membrane of the mitochondria. So we've got 3 carriers that I'll talk more about in a bit, okay? But that's why apparently there are 3 carriers. Now these are some of the players. I'd only ask that you be able to recognize them. You're never going to draw them. Leninger here has made in pink or magenta. They're identifying all the conjugated double bonds, single bond, double bond. And so you can see that with lots of conjugated double bonds, the rule of thumb in physics is that things that have lots of double bonds absorb light at higher wavelengths. Well, that's up in the visible range, not down in the ultraviolet. And so this thing is good at absorbing light if it has a metal ion. And that metal ion needs to be magnesium. I wish they had labeled this as magnesium +2 because that's what it has to be. So there's a metal ion here. This structure is reminiscent of the structure of a heme. It's a little bit different, but remember hemes have irons in there. This needs a different metal, a magnesium. So chlorophyll A has this structure with a long hydrophobic tail. And when there's a magnesium here, this molecule is part of what can absorb photons of light. Sometimes the magnesium is missing, in which case it can't absorb light apparently, but it can carry an electron because these conjugated ring systems still have the ability to share an electron without forming a nasty free radical. And so we'll find that sometimes these molecules, if they don't have a magnesium in them, they're still part of the electrical circuit. Just like in the electron transport chain of mitochondria, there's an electrical circuit. There's an electrical circuit here in the chloroplast, okay? So you need things that can carry one electron on its way. These things down here, beta carotene and lutein, have a very different structure than up here, so it's not a surprise that beta carotene, a compound that you find in carrots, right, absorbs light at a different wavelength, and so does this compound here. So these two compounds are referred to as accessory pigments. They can absorb light at wavelengths that chlorophyll A. Chlorophyll B is very similar in structure, except that there's an aldehyde group up here at the top. The point is, the chlorophylls A and B absorb a lot of light. That's great, but these guys absorb a lot of light at wavelengths that the chlorophylls A and B cannot. So that's nice to have these accessory pigments because organisms are trying to grab as much light from the sun as they can. It's a dog-eat-dog world out there. If imagine you're in a jungle, the light's pretty easy to grab if you're up at the top of the canopy, but down below you're getting whatever filters through. And so you're thankful for whatever photons of light were missed by everything else. So this is just a plot of, in general, the light coming from the sun. And let's take chlorophyll A here. It's very good absorbing light out here and down here, but not so much in the middle. And so things like lutein and beta carotenes and some other pigments can absorb light at those other wavelengths. So we're not memorizing this. The point is different compounds have different structures and hence can absorb light at different wavelengths. Okay, so this is where it gets complicated. And I don't have an easy answer, okay? So this is saying in Leninger here, they're taking a segment of one of these membranes in the thylakoid and saying, here's what it kind of looks like. And here is something, a protein complex called an RC, a reaction center, okay? So in this analogy that I'm going to make here, I'm the reaction center, okay? What's special about me? I've got two chlorophylls. I'll use my hand here to represent the porphyrin ring of one chlorophyll. I've got another chlorophyll, so I got another porphyrin ring. They both have magnesiums in them, okay? But they're positioned, all of this is mostly protein. They're positioned by proteins perfectly to be so close together they act as one. So this is reminiscent of complex 4 in the mitochondria, where we had two binuclear sites. Here is something, we don't call it a binuclear site, we just call it a chlorophyll dimer. So two chlorophylls act as one, and I have the ability, whereas you guys don't, to pop an electron up to a higher energy state. So in this diagram, I'm this reaction center here, and you guys are everything else, okay? So you're all these other pigments, chlorophylls, carotenoids, etc. Of course, there's lipids making up the membrane here. But basically, you're feeding, like a radar dish, you're feeding your energy to me. It's very efficient. And I'm constantly popping one electron, that's all I can do, one electron to a higher energy state. So let's think about that for a second. If I pop an electron to a higher energy state, then an instant later, a millisecond later, I'm devoid of an electron. Something has to feed an electron to me. That's not you guys. Something else needs to give me an electron so that I go back to my ground state and I can pop another electron up. Are you okay with that? So that I'm getting constantly fed electrons. You guys are feeding me what we call excitons. Excitons is an interesting term for something that we don't truly understand. Somehow it's at the quantum level where nothing really makes sense. In quantum mechanics, somehow you guys are transferring your energy to me. We think it might involve some quantum tunneling, but a lot of it is basically we don't know. I wish we did, but currently we don't know exactly how that happens. Is that okay? But I can, this is not, it just died. This is not quantum mechanics. Okay, we're good. Okay, so photons are striking you guys. The energy of that photon is being translated into an electrical wave. And that wave of energy is coming to me. And I'm using that energy to pop an electron to a higher energy state. And by doing so, on the y-axis, which is free energy, if I move something to a higher energy state, then as that thing drops down through an electrical circuit, it can be made to do work. Okay? So, my best example of how you guys are feeding energy to me, when I say we don't know, that's true. I don't know the fine points, but I can give you the general idea. And that would be, let's imagine it was a couple years ago when life was more normal here, pre-COVID, and we'd have clicker questions. I always hate clicker questions because half the class has no idea what I do, so I do, half of it's wrong. But it's a good example here because I would have a little electrical device with an antenna, and you guys would all have your remote controls, correct? And when you flash an answer to something on the screen, I get an instant response because it's coming out as a wave from your device. And that's how it's happening here. These are waves coming from each of these pigments. Far more efficient than it was a particle that just randomly walked. It would take forever to get to this device. Do you follow me? So it's a wave action that's coming to me. That's the best I can do, okay? So that's what's happening at this reaction center. I am gathering all the energy, excitons. Exciton is just a term for a quantum of energy. The energy came in as a photon from the sun, and you guys are transferring that quantum of energy to me. And it's very efficient because it's coming as a wave somehow to me. All right. So now let's look at this. These are the purple bacteria and the green bacteria I mentioned on almost our opening slide. We're not memorizing this, but I need you to take a step back and see what's going on, okay? So here on the y-axis, we've got energy in the form of the standard redox potential. That means everything's at 1 molar except for protons. Protons will be at pH 7. And so let's just focus with the purple bacteria. They have a reaction center, RC, pH 70. P stands for pigment, its maximum absorbance, that is the wavelength that it absorbs most in a spectrophotometer is at 870 nanometers, so imagine that's me. It's got a chlorophyll dimer. It can pop an electron up to a higher energy state. So when it does, the higher energy state is called P870 star. So this pigment is at a higher energy state and it can give off 1 electron. And 1 electron goes through the protein part, and thankfully, as scientists, it goes through pheophytin. Pheophytin is a chlorophyll without the magnesium. And so when an electron is going through a protein, usually we can't see where it's going. But when it goes through something like pheophytin, or here, your friend coenzyme Q from mitochondria, we can see a spectral shift. And then it goes to the cytochrome BC1 complex, which you're not memorizing, but this is the thing that is similar to plants and bacteria, mitochondria, the 2 caverns. This is where the Q cycle is occurring. That electron is carried by cytochrome C2, which is a small protein that is similar to cytochrome C in the mitochondria. And this sends this electron back to the reaction center so the whole thing can go again. Is that cool? So these bacteria, when they're harvesting the energy of excitons coming from the sun, can pump that electron through this cycle. Well, the cytochrome BC1 complex, just like complex 3 in the mitochondria, remember that's the 2 caverns, 1 risky iron sulfur protein goes up, then it comes down, and the other one goes up, etc. And every time it goes up, an electron gets moved, but then protons get dumped off. It's this guy that's creating a proton gradient that's going to run the bacterial's version of ATP synthase to make ATP. So as long as the sun is shining, these bacteria can make ATP. Cool? Okay, now let's go over here. These green sulfur bacteria can do the same thing, but also one more thing. And that is here, these bacteria have a reaction center called pigment 840. It's a different organism, so not surprising that it has a different maximum absorbance because it's held by different proteins. When excitons transfer to this, it pumps an electron up, which can come down and back, just like over here, or the electron can go to a protein called ferredoxin. I'll give you the spelling on another slide. And ferredoxin can feed electrons to an enzyme that reduces NAD to NADH. So these bacteria have a choice. They can either make protons that make ATP down here, or they can make reducing power in the form of NADH. Plants do not make NADH for this reason. The way that's being done here. They make NADPH. Okay? So if the electron winds up on an NADH, by the way, how many electrons does 1 molecule of NADH hold? 2. So this process would have to happen twice to make 1 NADH. So the NADH can, in this bacteria, diffuse away and be used to build something. Are you okay with that? If the electron disappears on an NADH, then not shown here is some compound has to replace the electron. If it goes in this cyclic thing, that's not a problem. But if it's being consumed, some other compound, well, in this case sulfur, has to be stripped of electrons to replace the electron that was bumped to a higher energy level. Cool? Okay, so we're not memorizing these things, but on the next slide, when we look at what happens in plants, it sure looks like what you learned probably years ago in biology, called the Z scheme, involves these two characters. So here is the Z scheme. You're not memorizing the Z scheme, but I need you to know who the components are, what they're doing, okay? What's the overall goal? So here, you see over here, photosystem 1, the electron can be pumped up. It can come back down this way to form protons, to form ATP, or it can go this way to form NADPH. This looks a lot like those green sulfur bacteria. This guy over here, instead of coming all the way back down, he's feeding into the other system. Okay? So it looks like two photosystems were coming from two different bacteria. And in evolution, somehow in evolution, some cell got lucky and incorporated two bacteria. And these eventually merged into what we call chloroplasts today. So this would be a process over billions of years. Okay? So let's tear this thing apart. P680 is this guy down here that we can barely see on the screen. If I'm P680, once again, I've got two chlorophylls in my reaction center acting as one, popping one electron up to this higher energy state. So we can say P680, you are a chlorophyll dimer. CHL stands for chlorophyll. There's two of them held by proteins so close together they act as one. Now, this is the ground state shown here on this y-axis. This is the energy that this complex has. When it gets excited by capturing a photon of light, or more likely from getting an exciton from one of the feeding groups in the radar dish, this gets excited to the chlorophyll dimer. That's this thing up here, P680 star. And then this thing can give off an electron. Pheophytin, again, is a chlorophyll without a magnesium in it. It's held by a protein. Everything is oriented perfectly by proteins. And this is the part that they're not showing on this diagram. When that electron comes off, so it forms CHL2 with a positive charge, so this is P680 plus, and an electron. That electron is coming off here. That's nice, but you can't forget the other part of the chemical expression. P680 plus is way off scale. It's way below this thing on this diagram at a positive 1.3 volts. So on this scale, it'd be down by the floor, okay? So what I would like you to remember, and I'll try and emphasize it as much as I can, remember when we had that table of redox potentials for mitochondria, and oxygen was at the bottom, and it wasn't even close. I said, oh, give me those electrons. That was like 0.8. So this is like, give me those electrons. It is the most positive redox potential known in biology. You're not messing around with P680 plus. It is so thirsty. It's like a black hole for electrons. And it's going to be sucking electrons from something called the OEC, Oxygen Evolving Complex. And this thing is stripping electrons from water, of which there's plenty, to give oxygen and then give back the electron that got popped up to P680 star. So life has an incredibly oxidizing compound called P680 plus that doesn't take no for an answer. It's going to grab the electrons from water. Okay? Another thing that we can say right here is that, and they don't show it here, but they should have, when you strip electrons from water, you're also stripping protons from water. So this oxygen evolving complex is a source of protons. Those protons, in addition to the ones coming from the cytochrome B6F complex, this is the one with two caverns, one arm going up, one coming down like this. There are two sources of protons here. And these protons are what is running the chloroplast version of ATP synthase. I need you to know about cytochrome B6F, okay? All right. So this thing is way off scale here, way down there. There's two guys here. PQA, as it says down here in this box, is plastoquinone A. The molecule looks very similar but not identical to coenzyme Q in the mitochondria. Its sister, plastoquinone B, is identical to plastoquinone A. The difference is plastoquinone A is held firmly by a protein. It does not move. Plastoquinone B, though, does. And when it moves, it carries one or two electrons, and it always carries them as H dots. So it can carry a maximum of 2 electrons, and when it does, it's carrying 2 protons. Are you okay with that? And what's cool about this, again, it has the structure of coenzyme Q pretty much. This thing is moving in the thylakoid membrane. It is mostly hydrophobic. If you're going to carry protons and electrons in the interior of a hydrophobic membrane, you better not have any charge. If you move them as an H dot, the net charge is 0. So everything is okay. So plastoquinone B, I'm putting a circle around you to indicate that you're a mobile electron carrier, and you always carry protons too. Plastocyanin, the cyanin part tells you that it is a protein that carries a copper ion. It's got a copper on it. And it can carry 1 electron. It does not carry any protons. So it's going to carry the 1 electron back to this photosystem here. Because when this photosystem pops an electron, P700 has a chlorophyll dimer. It pops an electron, and when it does, it better get it back. Plastocyanin usually is the source that provides that missing electron. So this guy goes to a higher energy state here. The electron goes down through these pigments, including an iron sulfur center. Here's ferredoxin. Ferredoxin is the 3rd carrier. Ferredoxin can only carry 1 electron, no protons. Ferredoxin is going to feed this enzyme, ferredoxin NADP oxidoreductase, to generate an NADPH, okay? A1 up here is a compound called phylloquinone. It has this structure. You're not memorizing this. The point is there's lots of conjugated double bonds on this quinone structure here, this benzene ring with a double bonded O, double bonded oxygen here, is held by a protein, and 1 electron can go through this thing, okay? So we're going to end today by talking about how many photons of light does it take to make 1 NADPH in what is called the Z scheme. Okay? So first thing we have to acknowledge is how many electrons is does NADPH carry? Anybody? Louder? 2, with confidence, 2. Okay, so 1 photon of light will move 1 electron from P700 up to here, down to give 1 electron, pardon me, yes, 1 electron of NADPH. So it took 1 photon of light to move 1 electron onto NADPH. Photosystem P700 can't do that again, though, unless an electron is moved to replace it. Do you follow me? So that means 1 photon of light has to come over here to P680, a different photon of light, because it has its own antenna. So now I'm up to 2. 2 photons. So 1 photon over here bumps an electron up, comes down, and replaces this one over here. So right now it's taken me 2 photons of light to get 1 electron onto NADPH. So clearly you're going to have to double that, so you're going to need 4 photons of light to make 1 NADPH, which is a piece of cake if the sun is shining. Okay? But I want you to see how the Z-scheme, the 2 photosystems, this is PS1 up here, this is photosystem 2, they are totally connected to each other. If one bumps an electron over, the other one needs to bump one over to replace it. Cool? That's enough for one day. We'll pick this up next time, and stay hydrated. I'll see you next time.